to have collated all this information. We were involved in learning about those genes from the very beginning. Several of them we discovered right here at Mass General and at Harvard. So we've made a lot of contributions. You've held out your arm to give blood samples, and we've used those blood samples for good purpose. Now, we always knew that it wasn't just what you inherited, but that there were other things that seemed to lead to these problems as people got older. And those are generally called risk factors. And so we've been studying those hard too. And yeah, we've got it. It's 2014, so everything's a website. So allsrisk.org is a website that Dr. Blacker is primarily responsible for. And it goes back and looks at all the world's literature, not just what we've done here, but every place in the whole world, and says, what is it about high blood pressure, or what is it about diabetes, or what is it about eating broccoli that either is good for you or bad for you? I can't say I'm a fan of broccoli yet, but even so, there's all this information that we've been gathering over the years, and that makes a big difference. And finally, the ability to just study individuals year after year after year. I frequently tell the subjects who I see in this study that each year we get almost twice as much information as we did the previous year, because we know about the previous year, we know about that year. And so think about that if you've been coming for 17 or 18 years, how many doublings that is, how valuable that is. I mean, it's so important, and ultimately, of course, we've asked almost everybody here to help us in that final study, which is to look at the brain after people pass away and just understand what is it that actually happened and how does that relate to what's going on. And that's led to some really fascinating studies. Dr. Gomez-Isla is here today in the audience, and she's leading a study in which she's looking at patients whose brains had Alzheimer's disease, but they didn't. They were great, even though the plaques and tangles that show up in Alzheimer's disease occur. Now, how can that happen? Well, Teresa's going to find out, and it's going to be because we've had the opportunity to learn those kinds of things. So 30 years ago, we knew that patients had memory problems as they got older, and that some people got Alzheimer's disease, but not everybody, of course. And we wanted to use that information to figure out how could we learn how to treat patients. And so I just want to introduce to you some of the members of the team we've been working with. This little picture, which represents DNA, this double-stranded helix. The team led by Rudy Tanzan here has been discovering those 20 genes that I told you about. These plaques and tangles occur in the brain, and the brain has neurons. The nerve cells in the brain die. Dr. Matt Frosch is the head of neuropathology now and at Mass General, and he's been leading the studies in understanding those brain lesions. We've been looking at the natural history of the disease, when the clinical diagnosis starts, and this kind of mapping out of the disease process led us to highlight these prodromal and mild cognitive impairment and even pre-symptomatic phases of the disease. And you'll be hearing from Dr. Risa Sperlin about some incredibly exciting work that's going on to try to understand what happens in the brain years and years and years before symptoms happen. Because we deeply believe that that's the right time to intervene. That if you fix problems before they occur, that's going to be an easier fix than waiting till a lot of destruction has already occurred. And it's understanding what's called the natural history, understanding the life history of diseases that's led to really the field wide rethinking about disease, the recognition that some changes occur years before clinical diagnosis. And it's really given Dr. Sperling the opportunity to launch the incredibly exciting A4 trial. And then there's some new things that we're really excited about. I mentioned to you that there are these things called tangles and plaques that occur in the brain, and that 30 years ago, the only way to see them was to ask a neuropathologist to look under the microscope and see them after someone passed away, and they had the chance to look at the tissue. And that's all changed. Now we can see those plaques. see the tangles as well. And so this is part of the reason why we now know that the brain years before people have symptoms, and it'll give us the opportunity to try to please. Neurofibrillary tangles, NFT, sorry. That's a TLA. A three-letter acronym. So this emphasis on early diagnosis and new therapies, I think, is something, a theme that's driving through the center. And many of you know Mark Albers or met him out front here. He's leading a group that's interested in the idea that there's some features of the sense of smell that might predict who's going to run into troubles. And this notion of the PET scan showing us the amyloid in the brain, of spinal fluid being able to give us biochemical indicators of things that are going wrong in the brain. Those are things that we've been taking the lead in and working hard on and trying to understand. And the whole idea is try to fix things before they run into trouble here. In the E4 trial that Dr. Sperling and Dr. Rentz are leading, you'll hear more about that in a few minutes. And then we've got a few projects that really dive deeper into the science. So in addition to these big programs in which you've all participated, there's some very specific things that are going on. We're trying to develop better animal models of disease in the laboratory. We're trying to develop new ways to do MRI scans in which we can take advantage of a brand new technology called Connectome in order to look more in more fine detail at the brain. And you'll in the next 6 months or so, we'll be asking you to visit with Dr. Hennon to sign up for those scans. And then I already mentioned Dr. Teresa Gomez-Isla's study of why some people are resilient to the disease. What is it that protects them? We sure want to know so we can all put some of that on our cornflakes every morning. Mostly what all of us want to do is just say thank you to the participants, the subjects, the patients who helped with the trials, and those of you who have come today just to help out and wear purple and celebrate with us the successes that we've had so far and work together with us as we look forward to trying to make more progress so we can do something about these things. So thank you very much.